Okay, this is the second part of lesson 3.05 on osmosis. We've got a lot going on in the middle of the table. Let's just ignore that for a second. And I'm going to draw some cells. So I'm going to start over here and I want to draw a cell. I'm going to put glucose again on the outside and on the inside. All right, so we're going to do our solute. Say it's glucose. And I'm going to put 10 dots on the inside. And on the outside, I'm going to put 4. Now, what you have to understand is that cells in our whole body, it's water everywhere. There isn't any place that doesn't have water. In fact, if cells don't have water, they can't live. So we've got water here, here. We've got water everywhere, but we're not really worried about the water. What we want to know is what direction is the water going to flow. So remember, we said in the last time, our solvent is water. And it will flow in such a way that it dilutes concentrated areas. Concentrated solute areas, if we're being more specific. So looking at this diagram and understanding that the red dots are our solute, and understanding the water has to try to flow to spread out the dots, what way is the water going to flow? Well, I have 4 dots on the outside. I have 10 on the inside, so the water is going to flow like this. We call this a hypotonic solution. Let me move this over a little bit. In a hypotonic solution, there is more solute inside the cell than outside. Well, you might say, but wait, the last example we had, the tube was open to the air so the water could move. Well, cell membranes, remember, they're fluid. They can stretch, they can bend. And so this can happen a little bit and we're okay. But look what happens if I have too much, if I put solution around the outside that has too little solute, we get this thing called lysed. Can you see what this cell is doing? Lysed means to burst. And by the way, these are all animal cells. So across the top, these are animal cells. So lysed is burst. And this can happen. If you got hooked up to an IV of pure water, so if you got hooked up, if they went down and got some spring water off the Walmart shelf and put it in an IV, this would happen to the cells lining your vessels because there would be so little solute in the IV fluid that it would flow into your cells, it would burst your cells. That's why when you get an IV, they make sure it's 10% salt. They put salt in your IV solution. You have sodium chloride in there, and that means that you have what's called isotonic. So isotonic means the solute outside and inside are equal. So the amount of water that comes in is equal to the amount of water that leaves. Hold on to this because we're going to talk about plants at the end. Well, can we go the other direction? Let's draw another cell over here on this side next to. What if I have 4 4 dots is solute, and around the outside I've got 10. Remember, we're not talking about the solute moving. We're talking about the water spreading things out and making it even. So do you understand how the water is going to flow in this scenario? The water is going to flow out of the cell to try to spread out these solute molecules. We call this hypertonic. For a hypertonic solution, there is more solute outside the cell than inside. Water leaves. Over here, water enters. So, okay, well, what's going to happen if our cells start losing all of their water? That's what we have here. We have a hypertonic solution. The cells losing water, they shrivel. When can this happen? Sometimes if people are stranded at sea, they'll get desperate and they'll start drinking salt water. Salt water has lots and lots of solute, and it creates a hypertonic situation. And they actually die because their cells shrivel up. Don't worry about this. We'll talk about that later. Now you'll notice for plants, plants can't handle hypertonic at all. And in an isotonic, we have this word which means flaccid. Flaccid means wilted. How do you stay on your, when you stand up, what is it that supports your body? Your skeleton, right? So plants don't have skeletons. What do they use to hold themselves upright? They have what are called cell walls. So this is a cell wall to a plant. And plants try to keep themselves in hypotonic solutions so that their cells are always swollen. They want that. They call that turgid. Turgid. Rewrite that. And they use that pressure to help the plant stay upright. So animal cells and plant cells react a little bit differently. What we're going to do is we're going to put some onion cells on a slide and add some salt water and add some fresh water, and we're going to watch them go back and forth between here. But you have to understand that you have to look at where the solute is and look at how the water's flowing to understand what's going to happen to the cell. I will tell you there is always at least 2 or 3 questions on the end of the year test about this topic. So if you watch, make sure, hopefully you watch this whole video. And at the end of the year, when we get ready for the end of the year test, I'll have you go back and watch it again. So it will show up quite a bit on the test. So our takeaways, we still have our solute solvent. In a hypotonic solution, there is more solute inside the cell, so the water flows in to dilute it. In a hypertonic, more solute outside the cell, the water flows out to dilute it. And you end up with a cell that potentially can burst or shrivel, depending. All right, that's the end of osmosis. And the next lesson will be on facilitated diffusion. Thanks for watching and have a good day.